I got broads in Atlanta.
Hello everybody. Today's biology video will be about meiosis. Meiosis contains phases interphase, prophase, metaphase, anaphase, all part 1, and then repeats it for part 2. In the beginning interphase, like mitosis, meiosis is preceded by an interphase, during which the chromosomes duplicate. At the end of this interphase, each chromosome consists of 2 genetically identical sister chromatids attached together. But at this stage, the chromosomes are not yet visible under the microscope, except as a mass of chromatin. The cell centromere has also duplicated by the end of this interphase. Prophase 1 is the most complex phase of meiosis and typically occupies over 90% of the time required for meiotic cell division. Early in this phase, the chromatin coils up so that individual chromosomes become visible with the microscope. In a process called synapsis, homologous chromosomes, each composed of 2 sister chromatids, come together as pairs. The resulting structure, consisting of 4 chromatids, is called a tetrad. During synapsis, chromatids of homologous chromosomes exchange segments in a process called crossing over. Because the versions of the genes on a chromosome or one of its chromatids may be different from those on its homologue, crossing over rearranges genetic information. The genetic shuffling produced by crossing over can make an important contribution to the genetic variability resulting from sexual reproduction. As prophase 1 continues, the chromosomes condense further as the nucleoli disappear. Now the chromosomes move away from each other, and a spindle starts to form between them. The nuclear envelope breaks into fragments, and the chromosome tetrads, captured by spindle microtubules, are moved toward the center of the cell. At metaphase 1, the chromosome tetrads are aligned on the metaphase plate, midway between the 2 poles of the spindle. Each chromosome is condensed and thick, with its sister chromatids still attached at their centromeres. Spindle microtubules are attached to kinetic cores at the centromeres. In each tetrad, the homologous chromosomes are held together at sites of crossing over. Notice that, for each tetrad, the spindle microtubules attached to one of the homologous chromosomes come from one pole of the cell, and the microtubules attached to the other homologous chromosome come from the opposite pole. With this arrangement, the homologous chromosomes of each tetrad are poised to move toward opposite poles of the cell. Like anaphase of mitosis, anaphase 1 of meiosis is marked by the migration of chromosomes toward the 2 poles of the cell. In contrast to mitosis, however, the sister chromatids making up each double chromosome remain attached at their centromeres. Only the tetrads, which are pairs of homologous chromosomes, split up. Thus, here you see 3 still double chromosomes moving toward each spindle pole. If this were anaphase of mitosis, you would see 6 daughter chromosomes moving toward each pole. In telophase 1, the chromosomes arrive at the poles of the cell. When the chromosomes finish their journey, each pole of the cell has a haploid chromosome set, although each chromosome is still in duplicate form at this point. In other words, each chromosome still consists of 2 sister chromatids. Usually cytokinesis occurs along with telophase 1, and 2 haploid daughter cells are formed. Following telophase 1 in some organisms, the chromosomes uncoil and the nuclear envelope reforms, and there's an interphase before meiosis 2 begins. In other species, daughter cells produced in the first meiotic division immediately begin preparation for the second meiotic division. In either case, no chromosome duplication occurs between telophase 1 and the onset of meiosis 2. In organisms having an interphase after meiosis 1, the chromosomes condense again and the nuclear envelope breaks down during prophase 2. In any case, meiosis 2 is essentially the same as mitosis. The important difference is that meiosis 2 starts with a haploid cell. During prophase 2, a spindle forms and moves the chromosomes toward the middle of the cell. During metaphase 2, the chromosomes are aligned on the metaphase plate as they are in mitosis, with the kinetic core of the sister chromatids of each chromosome pointing toward opposite poles. In anaphase 2, the centromeres of sister chromatids finally separate, and the sister chromatids of each pair, now individual daughter chromosomes, move toward opposite poles of the cell. In telophase 2, nuclei form at the cell poles and cytokinesis occurs at the same time. There are now 4 daughter cells, each with the haploid number of single chromosomes. If you found this video on meiosis helpful and wish to learn more about different biology topics, please feel free to subscribe. This is brought to you by PHU Biology.
Going out like a Montana, honey killers on the helmets, legacies, found them, way see, pockets full.